highlights a problem in the bottle. In this particular case, wine was returned from clients 18 months post-bottling, and they were complaining about bready type aromas coming from the wine. And so then when the wines were analyzed, they definitely detected Brett and elevated levels of volatile phenols at ETS labs, and the data are presented here. And so you can see when the wine was bottled and checked the first time, and then when it was checked the second time after the complaints. And so you see pH, and you see the relatively low sugar content, the consistent ethanol content. The molecular SO2 is obviously very low and below antimicrobial, but even still, Brettanomyces is pretty resistant to normal levels of molecular SO2. But what's key here is that this massive rise in phenolics, the 4EP and 4EG, in that time frame. Without even looking at the microbiological data, this suggests that it's a bloom of Brettanomyces, or possibly Pichia, or one of these other strains of yeast that can also make these phenolics. But the confirmation is obviously here when you're looking at the molecular approach scorpions and the traditional microbiology plating data. Now, what's interesting about the case, too, is that the winemaker was mostly keyed into this data point right here, the traditional microbiological approach showing no growth of Brettanomyces at the bottling stage, but didn't appreciate the importance of this piece of data here, the highly sensitive molecular technique called scorpions that detected this low level of Brettanomyces, and still they bottled, or rather shipped the wines, knowing that this was there. Probably, obviously he took a risk and said, oh, this is more important than this. And in fact, as you can see, it was a terrible miscalculation. And so the levels of Brettanomyces detected after this nearly 2-year time period were very significant, and obviously enough to cause sensory problems in the wine and have individuals want to return the product. Another thing that I would point out is that plating often underestimates the numbers of microbes. So in this particular case, 71,000 versus 83,000. So there's a pretty big discrepancy there, well over 10,000 cells per mil. And one of the reasons for that is this technique here, the molecular technique picks up both live and dead cells, whereas this technique only picks up live cells, obviously. But there's another thing that can happen here, and that is this technique, the plating technique, will not pick up a third type of cells. So there's live cells and dead cells, but the third type is the intermediate form, which is the viable but non-culturable type of cell, meaning that this is a group of cells that physiologically are still alive, but they are unable to grow on traditional nutrient media. And so that's the group of organisms that we really need to be aware of, because they can still cause spoilage and they can still bloom in a bottle. And so that may be what happened here. Again, these guys, this plating did not pick up this small population of cells that the molecular did. And so again, that concept of viable but non-cultural becomes important and is something that we always need to be thinking about. So again, the client was used to plating data and decided that the Brett number by scorpion was not a risk, and obviously that came back to bite him in the butt. The low levels of sugars don't provide protection against Brett. To be honest with you, Brett can actually metabolize really low levels of carbon sources, and you can even switch to eating ethanol, which is pretty amazing. These are incredible survivors. And so it's not surprising that the yeast could do what they did. And so again, assume the worst in this particular case. If you see that low level in your scorpions, then assume that they're going to bloom. And obviously, you assuming the worst is going to help you in the end most of the time. If Brett is detected at low levels, then you can sterile filter or use DMDC or Velcorin, and I'll talk a bit about those. So again, Practices that can increase the risk of spoilage, obviously lack of sanitation, flirting with wines that have an elevated pH. There has been a bit of a call from the consumer to for slightly higher pH wines that are a little less tart, I suppose, and obviously underestimating specific risk in the various processes that you use, and that can include a whole bunch of different things. And lastly, what ETS Labs always talks about is reactive versus proactive winemaking. If you're proactive, you're doing all the appropriate wine chemistry and microbiological analysis. If you're playing reactive, then obviously you're just reacting to terrible things that are happening down the way. Some of the recommended practices, obviously, minimizing the load of spoilage microbes in your facility. So just keeping things clean, which is kind of a no-brainer, I suppose, but you'd be surprised. Screening for the presence of spoilage microbes by both plating and scorpions. Some people are just jettisoning the plating altogether, realizing that the plating isn't sensitive enough in many cases. And then obviously proper sulfite management, and certainly looking at things from the microbial perspective, which I'll talk about. So, again, checking the boxes of sanitation and making sure your barrels are clean, and cleaning and checking the condition of equipment, and certainly checking incoming wine, because you can bring in spoilage microbes from when you're importing wine from other wineries and doing blends and so forth. And so that's obviously hugely important. And then, of course, reducing the airborne load, which is a often overlooked process. Individuals rarely sample their air, but there's no question that yeasts are floating around, and all kinds of bad things can be in the air, molds in particular. So some of the recommended practices: screening for the presence of microbes, like I talked about. Just looking at your juice under the microscope is an important thing. You can tell a lot about it if there's a high level of gram-negative rods, for example, suggesting that acetic acid bacteria are coming, or unusual shapes of yeasts that are not Saccharomyces. And certainly plating, again, direct plating or filter plating, which I'll talk a bit about, are really important. But again, losing ground to scorpions, which is on the right here, which is more the molecular approach. Scorpions can not only identify the microbes, but specific microbes, but it can also quantify them. So again, recommended practice is obviously the most important tool. Well, I wouldn't say most important, but one of the most important tools is effective use of sulfiting, finding out the optimal molecular SO2 and then managing it. So the basis for its operation looks something like this. I mean, this is a really simplified diagram, but we typically use inorganic ions like this to inhibit wild yeasts and molds and bacteria. And so the idea is that you would use, often it's just powdered metabisulfite, which releases sulfur dioxide, and then these compounds can readily diffuse across the cell membrane in an acidic environment. And so once they get into the cell, which has a relatively higher pH, more of a neutral environment, these sulfur compounds ionize basically, and you form all kinds of radicals like sulfites, for example, that are highly reactive, and they can just cause a mess inside the cell. They cause all kinds of damage and acidification. They destroy DNA and proteins. So it's basically just unleashing a bull in a china shop. But the key here is you need an acidic environment, because if these chemicals ionize outside of the cell and form ions in the surrounding medium, they won't be able to penetrate the lipid bilayer of the cell membrane. So again, it has to be acidic to keep these molecules together from ionizing, and then once they get in, they can ionize here and then cause all their damage. So the big question of how much SO2, what's the optimal amount, and that's the goal is obviously to maintain the highest level without causing any negative sensory impacts. So that's something that obviously you may want to determine empirically. There's a huge pH pitfall, which I don't know if a lot of people know, but in wines with high pH, it's possible that you won't be able to reach an SO2 level that's high enough to prevent microbial growth. And so I put an example here just to see what I mean by that. So to achieve a molecular SO2 of 0.8, which I talked about being pretty good antimicrobial level, a pH 3 wine only requires 13 ppm of free SO2. Whereas to achieve a 0.8 in a 3.4 wine, you need 32 ppm. So again, it's very pH sensitive. And again, this is why, right? This is what I was just talking about. The lower the acidity, the more deadly this is. I'm sorry, the higher the acidity, the more deadly this is. The lower the pH, the more deadly these guys are. And so this is just a silly little graph looking at free SO2 on the y-axis in time, and showing you here's our target. We want to hit 0.8, for example, and this is what happens in a lot of this is the oscillation that typically happens in a lot of wineries, and this is probably ideal. This is probably the best we can do in terms of the ups and downs of SO2 levels, given all the things that can happen to SO2, binding to solids and other chemicals that interfere with it. But this is what happens in most wineries in terms of the fluctuations in SO2. And each time you get one of these really severe troughs here, these are periods of time when spoilage microbes can take off and cause problems. So looking at the microbial perspective, obviously we've got to look at the wine chemistry, the presence of inhibitors and so forth, and how those would impact microbial growth. And so again, growth promoters, what's the sugar content, the malic acid, the YAN, all these things, and are they too high? Are they too low? And so again, they have to be adjusted so that if they're too low, you may choke out your primary yeast and allow weeds to grow, not literally obviously, but weed microbes to grow. If it's too high, then everybody can grow, so you've got to find those sweet spots. And obviously the presence of growth inhibitors are key to managing the microbial populations as well. And so some of the approaches that we can use, preservatives and sterilants, we can use organic acids like sorbic acid, or even lysozyme, which is an enzyme that you and I produce. It's in our tears and saliva, but it's it's even in chicken eggs. It's a pretty common enzyme that breaks down bacterial cell walls. And then valcorin, or DMDC, which I'll show you in a minute. So sorbic acid, basically has the same premise of most organic acids, or all organic acids, in terms of their preservative effects. We use, for example, acetic acid and organic acid in mustard or ketchup. And they all work basically the same way. Here's your organic acid, and because it's not ionized, it can move into the cell and ionize in the cell. This proton that you see here, H+, proton, will acidify the cytoplasm and cause all kinds of problems, damaging enzymes and lysing DNA and so forth. And these anions that accumulate can also damage DNA and protein function. And so again, the accumulation of these counterions, these anions, and then these protons can cause all kinds of internal problems. The other thing that these things do, though, is, by acidification, that causes a stress response in the cell. And so the cell is provoked to take those protons and pump them out of the cell. But notice what's happening here. The cell has to use energy in order to pump that proton out of the cell. So this is energy that would be devoted towards building things or motility or something useful that now has to be allocated towards survival. And so this cell is no longer using its excess energy to grow. It's actually using the excess energy to mitigate the acid damage. And so that in and of itself may not kill the cell, but would keep spoilage microbes from overgrowing. The potential problems are shown here. They don't have a very long active period, these organic acids, and there's a potential to add off flavors. Sorbic acid can produce ethyl sorbate, which has a bit of a anywhere from pineapple to celery flavor. I've heard, aroma and flavoring experts, detect both things, off flavors. And again, they're not highly effective against certain lactic acid bacteria, but for sure gram positive, and for sure some gram positives and some gram negatives, but not all of them. Now, if you want to get rid of gram positive, primarily gram positive lactic acid bacteria, you can just add lysozyme. Lysozyme is a bacterial cell wall degrader. And so again, a lot of these particularly gram positives with their big thick cell wall are very susceptible. And so again, you can see this. Here's 2 test tubes in which you've added a turbid suspension of bacteria, and then you add a slug of lysozyme into one, pictured here, versus one that doesn't, and then contain lysozyme. And you see a bit of turbidity in here, but the lysozyme in this particular case basically just chopped up all the cells and eliminated them. So again, here's the cells, pre-lysozyme treatment, then you add the lysozyme and they just blow up. So lysozyme can be added directly to wine and mixed around a bit and doesn't cause any taste or visible issues. It'll largely just dissolve in solution, and it's totally natural. Like I said, it's present in our saliva and our tears, and it's present in chicken eggs and stuff. So you wouldn't get in trouble for adding some weird chemical if there are people out there that are highly into organic products. Velcorin or DMDC, dimethyl dicarbonate, is typically also used. And so DMDC is really good at attacking enzymes that have an imidazole group. So this is imidazole groups are present in typical particular amino acids like histidine. So any amino acid or any protein rather that contains the amino acid histidine would be highly susceptible to DMDC. And so again, this molecule gloms onto them, binds to them, and damages the enzyme, so it doesn't work, and that causes enzyme inactivation and thus cell death. And so, a typical thing that you could do is something called like the Velcorin test. And here's the Velcorin, and you can add that to a bottle, crack the bottle open, add it to about 200 ppm, and mix it up, and then run the contents of that. Let it, expose the Velcorin to the wine for a period of time. It could be very brief, and then dump it through a filter apparatus. And then you can take the filter off and then place that onto some microbiological media, and then count any residual microbes. So obviously you do it before and after. So you could do it this way, where you take a bottle of your wine and crack it open and run it through the filtration system. Then you can take a separate second bottle and add Velcorin to it, let it sit for a period of time, and then run that through the system, and then you can compare. You can compare the two to see, number one, what's your load of microbes in a bottle of wine, and then did the Velcorin clear out the spoilage microbes? So in conclusion, the main things are basically just developing a plan to monitor, and usually that means chemistry and microbiology. And so the end result is that it's better to be data driven and make decisions based on data than get any unpleasant surprises.